Hi, this is Yosuke Tanigawa, and I'm a graduate student at Stanford University. I'm excited to present our work titled WhichTF is functionally important in your open chromatin data. We have nothing to disclose. A preprint describing this presentation is available on bioRxiv. Please check the link in the description. Transcription factors, or TFs, regulate cell type specific functions. If you're interested in finding the functionally important TFs, what would be the possible approaches? One thing you could try is to run experiments such as DNA-seq or ATAC-seq to characterize the open chromatin region as the proxy of the active cis-regulatory elements. Let's consider what computational approaches are available to identify TFs given such measurements. As an example, let's say you're interested in the transcription factor acting in T cells. Once you have the ATAC-seq peaks, you would expect the reads from the accessible region have enrichment for the sequence motif of the active TFs in the sample. Luckily, there are many tools that can compute such abundance-based enrichment. Let's see what's going to be the top-ranked TFs from commonly used tools. Applying MemeChip from MemeSuite to publicly available DNA-seq data characterized on T cells, we identify the top 5 TFs. Unfortunately, the results are not exciting because regulatory factors involved in housekeeping processes dominated the top hits. For example, CTCF is a commonly known structural element that shape chromatin loops in many cell types. What we want as a biologist is a TF with cell type specific functions. Next, we considered applying enrichment analysis using predicted TF binding site in conserved regions. To that end, we applied PRISM, an algorithm previously developed by the lab, and performed the enrichment. Again, CTCFs and other housekeeping processes dominated the top hits. What if we look at the accessible TF binding site present in the enriched function? Applying genomic region enrichment of annotation tool, or GREAT, we can find cell type specific functions enriched in your accessible regions. Briefly, this tool considers ontology-based gene-level annotations for putative regulatory domains. Since its original publication in 2010, GREAT has been used for more than 1 million times and cited for more than 2,000 times. When we apply GREAT to our T-cell example, we find many T-cell related functions such as abnormal T-cell differentiation, activation, and proliferation. In WhichTF, our proposed approach, we focus on the TF binding site associated with those functions and ask if we can find cell type specific TFs in action. Here, we have the top ranked TF from WhichTF for T cells. The top ranked TF, NF-kB1, is a TF in NF-kappa B signaling pathway. The cell type specific function of this NF-kappa B signaling pathway is well established in literature. Similarly, ranks 3, which is our second ranked TF for T cell, is also known to be involved in T cell development. Overall, the cell type specific function of the identified TF are supported by literature. So far, we show the result for T cell as an example, but we demonstrated the ability of WhichTF in many different contexts. In our preprint, we present the result for both human and mouse datasets, samples along with developmental trajectory, as well as disease-related samples. Some of our test data are from DNA-seq experiment, whereas others comes from ATAC-seq experiments. When we look at the literature, a lot of top-ranked TF have support for their cell type specific functions. We also have many examples of novel discoveries, especially in under-characterized samples. To analyze pair of closely related samples, we have developed a differential analysis framework and validated its utilities. WhichTF identifies ranked list of TF as well as its score. We show that the quantitative score used in the ranking are useful to project cells into lower dimensional space. If you're interested, please check our preprint on bioRxiv. You are more than welcome to reach out to me using the conference platform, Twitter, or email. Thank you very much for visiting our presentation.